I'd now like to introduce Professor Tom Snelling as our next speaker. Tom is the Director of Health and the Clinical Analytics team in the School of Public Health at the University of Sydney. His pioneering work in the application of Bayesian approaches to design, coordination, implementation and analysis of public interest study, and is successfully leading a suite of multinational collaborative learning health projects across Australia. So without any further ado, I'd like to welcome Tom Snelling. Thank you, Tom.
Thanks, Sally. This is the COVID-19 crisis. I'm just going to talk about trying to resolve uncertainty with regards to pandemics and how we're doing with COVID-19. So you'll know that as of today, we are up to 10 million cases across the world confirmed to be infected with COVID-19. Almost certainly an underestimate of the true total number of cases, and concerningly, over 500,000 of those cases have died, and even more concerningly, the rate of deaths is continuing to increase in a fairly linear fashion, and it's not actually clear that we are doing any better with managing COVID-19 cases than we were 4 months ago. This is not only having effects, obviously, on health systems around the world, but you will be aware and appreciate the significant impact that this is having on economies around the world. So we've been working on a project trying to identify the key areas of uncertainty, specifically with regards to management decisions for individual patients infected with COVID-19. Obviously, there's a whole lot of uncertainty and questions around management of the COVID-19 pandemic from a whole of population level, but our focus has predominantly been looking at questions regarding the optimal management of individual patients. And so this diagram sets out a typical journey of a patient with COVID-19 and the sorts of questions that arise for clinicians who are caring for those patients at different points in their journey, and what is the sort of information that we might have access to in order to resolve that uncertainty. The key questions really for patients is, one of the key questions is around where they should be best managed. So a patient who is confirmed to be infected in the community, the first question with someone who has a nose swab for COVID-19 is, can the result of the nose swab be trusted? Because we've had problems with both false negative and with false positive results. But assuming that someone is infected, then there are further questions about whether this is a patient who can be safely managed in the community, or is this a patient who needs to be admitted to hospital? And if they are admitted to hospital, then there are further questions about if and when they are safe to be discharged to home, or are they at significant risk of death and need actually to be in the intensive care unit. And again, when they're in intensive care, a whole range of questions about the optimal management of those patients, not just about where they should be managed, but the treatments that should be administered at the different time points of their care. So as you can see, there are significant and important questions at all aspects of the patient journey that need to be answered. But the problem that we have are the tools that we have that are able to address those questions. And the title of this paper is The Answer is 17 Years, What is the Question? And I can tell you that the question is, how long does it take on average for clinical research to be translated into clinical practice? And the answer is about 17 years. And that has to do with all sorts of inefficiencies in the way that clinical research is conducted, and inefficiencies in how conclusions from clinical research are actually translated into changes in practice. So we could undertake a series of conventional clinical trials and research projects if we wanted to impact on patient management in 2037. But if we want to impact on the management and outcomes for patients who are infected today, then arguably we need to be doing things differently. And of course, there is a lot of research effort. And the question is, why is it that with so much research effort going into COVID-19, why are people still dying at unacceptable rates? And why is it that our management is slow to improve, despite so many deaths and so many patients being affected? Part of the problem is that our clinical trials and clinical projects traditionally operate in silos. The processes for conducting clinical research are burdensome and time-consuming, and this particularly affects frontline staff who are providing care, and they're having to divide their time between providing care for their patients and also providing study-specific procedures to address some of these questions. Typically, it takes a long time to be able to reach an answer. Many of the trials, in fact, most trials that are conducted are inconclusive at the end of the day and don't actually result in any outcomes that can be implemented in practice one way or the other. And so what we have is a information void, and we have practice that is occurring within this information void. And this results in a whole lot of crazy practice going on. And we see people, prominent people in that void getting into the media and other places and advocating for specific treatments in the absence of any evidence. And that can be for relatively sensible things like hydroxychloroquine. But we've also seen some quite crazy things that have been proposed as treatments, including intravenous bleach. And one of the motivators for this is this idea that when you've got nothing else to lose, then you might as well try anything. But the problem is that, of course, you do have a lot to lose. And that's not only loss if you're harmed by a treatment that is ineffective, but there's also a significant opportunity cost as well if you end up having a treatment that doesn't work at the expense of a treatment that might. And so these are all this is a snapshot displayed in a network diagram of the various treatments that have been proposed for treatment of COVID-19 and that have not only been proposed but are actually in clinical trials or clinical studies of one form or another. And as you can see, for a number of these treatments, that there are often multiple trials that are being conducted, and often those trials are individually small and unlikely to be conclusive in their own right. So an exception to this and of great excitement has been the recovery trial. The recovery platform is a UK-based platform. It's a multi-arm study, so they're not just looking at a single treatment. They're looking at a range of plausible treatments. These are treatments which are being repurposed, so they're already licensed for use for other indications. It's been postulated that they might have effectiveness for COVID-19. They're relatively safe and well tolerated and so they have entered into this large platform trial. So the specific treatments that they've been looking at, one is a repurposed drug used for HIV, which is an antiviral therapy, which has been suspected to have efficacy for COVID-19. One is a steroid or an anti-inflammatory medication. Hydroxychloroquine received a lot of press. That is an anti-malarial agent, also with some immune modulating properties that's been proposed as a treatment for COVID-19. And the other, azithromycin, is an antibiotic, which also appears to have some immune modulating properties and also has been proposed as a treatment for COVID-19. But the significant features of the recovery platform compared to traditional clinical trials, firstly, the consent form is only 2 pages. That compares with a consent form for most clinical trials that usually run over 10 pages or more. A single ethics committee approved the protocol for all of the UK. It took them 3 days to draft and finalise the master protocol, 5 days to have the protocol approved by the ethics committee, 9 days to have their first patient recruited, 24 days they had 1,000 patients enrolled, and as of today they've got more than 11,000 patients recruited. So this is the way that we want to be doing clinical trials. And you may have seen that in the last couple of weeks, that trial is now starting to bear fruit. And so we have the report just a couple of weeks ago that dexamethasone, which is a steroid treatment, is actually shown not only to be effective, but to prevent death in patients who are infected with COVID-19. So on the 5th of June, they reported that hydroxychloroquine, the treatment that was widely touted by Donald Trump, was not effective. 16th of June, they showed that dexamethasone reduced the risk of death, and in the last couple of days, they have reported convincing evidence that lopinavir, ritonavir, which is the HIV medication, is of no benefit to patients with COVID-19. Of course, some of the devil is in the detail with regards to dexamethasone and its effects. If you look at all patients, there is a modest protective effect. So this is looking at the risk of death over 28 days. Patients who receive dexamethasone are less likely to die than those who received usual care without dexamethasone. And that benefit was even greater in the subset of patients who are very severely unwell and requiring mechanical ventilation in intensive care. But interestingly, patients who are not severely unwell and in fact not even requiring oxygen therapy in hospital, there was a suggestion that patients who received dexamethasone may have actually done worse than those patients who are receiving the standard of care, which is of concern. And if you look at, you can split the data up multiple ways, and you can see that there is a suggestion that not everyone benefits from dexamethasone. Patients who are younger appear to benefit, and patients who have been sicker for more than 7 days appear to benefit. This is a forest plot where anything on the left-hand side is suggestive of a positive effect. Anything in the middle or on the right-hand side is suggestive of either no effect or potentially a harmful effect. So really the question going forward is not whether dexamethasone is beneficial or not, but in whom is it beneficial? And we need to have trials that can start to drill down on these issues. And so we're starting to evolve this causal model which suggests that in the early parts of disease, their problem is uncontrolled viral replication, which makes you ill, but the people who really run into problems are those who control the viral host the viral replication, but then have a significant and uncontrolled inflammatory response, immune response to the virus, and so different treatments are likely to have effects at different stages of this disease. And the important thing is that suppressing someone's inflammatory response with steroids may be beneficial for those with severe disease, but is unlikely to be beneficial at those in the early stages of disease who don't have a heightened inflammatory response. And in fact, it may be deleterious, and that appears to be as though that may be playing out in the data. And so our group has been working on these causal models, more explicit causal models, to try to understand the pathogenesis of COVID-19 and to help to design interventions. And these models now are starting to grow in their complexity, and they've been developed through a process of expert knowledge elicitation. And we use these models then to help us to design clinical trials to evaluate which treatments are effective and which are not. So how can we perform trials better suited to pandemics? Firstly, we need to have shared platforms for enrolling and capturing data and for a synthesis and analysis of data. We need to be able to evaluate multiple candidate therapies simultaneously, not in a piecemeal way. We need to be able to add interventions when they're available and evaluate them as quickly as possible. We need to be able to fail quickly, so we need to be able to drop any futile therapies as soon as possible to give way to more promising treatments. And we want to be able to focus enrolment, that is to allocate more participants to more promising therapies. And then we want to be able to implement and disseminate these findings in a timely way. And the way that we can do this is through what's called a platform trial. And so a platform trial is, the analogy is that the modern sports arena for clinical research. The idea is that you don't want to build a sports ground or a sports stadium for every game, for every study. You just want to build it once. You want to apply consistent rules. You want to allow multiple teams to compete, that is multiple therapies to be evaluated. And you also want to be able to allow multiple sports to be conducted there, or evaluate your therapies in multiple patient groups. And so the idea of a REMAP platform trial is that they are randomised, they are embedded in healthcare, they're multi-arm, they're adaptive, so they allow for changes to be made over the course of the trial, and they are pragmatic, meaning that we try to make these as efficient and as applicable to real world as possible. And this is typically what we have. We don't have a single type of patient and a single intervention we want to evaluate. We've actually got multiple interventions and we've got multiple types of patients, and we're trying to look at the effectiveness of each intervention in each patient. And we do this, in order to be able to do this, we can't use traditional fixed designs. We need to use these adaptive designs where we set specific rules, but we set those rules to allocate patients as efficiently as possible to the most promising treatments. And I don't have time to go through all of those details, but we do have a website that people can find their way to, and we've got a series of videos and materials there that you can look at. So just in summary, platform trials are really built to resolve uncertainty as quickly and as efficiently as possible. They are particularly important for pandemics, but they shouldn't just be for pandemics. This is how we should be doing clinical research for everything. they should be set up to be perpetual. So the idea of having a single standalone trial to answer a single question is very inefficient. We should be setting up platforms that exist for as long as there is uncertainty and wherever there is uncertainty in clinical practice. They offer faster knowledge gain and implementation of that knowledge. They are arguably more ethical, and they're ethical if they're designed in a way that allows us to allocate patients to the most promising treatments, which means that you're optimizing the treatment of participants, not just trying to discover results for future patients. On the downside, there are complex data and digital infrastructure requirements, more so even than for traditional trials, as a significant amount of effort up front and lead time in order to design these platforms through simulation. But in order to address this, we shouldn't be waiting for pandemics. We really need to start now to establish and create these platforms, establish the infrastructure and the governance frameworks to allow us to implement these platforms as quickly as possible. Thank you.